If you've ever felt like a heavy wave of brain fog right after eating a massive, super rich meal, you've actually experienced this tiny temporary fraction of the gut-brain axis at work.
Yeah, that's exactly what it is.
Right. It's that sluggish feeling where your thoughts just seem to be moving through molasses. But consider what happens when that system doesn't just slow down, but it catastrophically breaks. Like what happens when the liver completely fails and the gut literally begins poisoning the brain?
It's a devastating cascade, honestly. And I think it highlights something we often forget. Our organs don't operate in isolation. We are this deeply interconnected ecosystem. So when a major hub, like the liver, goes offline, the collateral damage just spreads everywhere.
Everywhere, yeah.
Especially to our neurology.
And that ecosystem is the focal point of today's deep dive. We're looking at a 2026 paper from the journal Cell by Agrawal and colleagues. It's titled Engineered Commensals for Metabolic Modulation of the Gut-Liver-Brain Axis. And the mission here is just fascinating. We're going to explore how scientists took common, everyday gut bacteria and basically program them to act as microscopic chemical factories.
Yeah, literally bypassing a failing liver to rescue a dying brain.
Exactly. Okay, let's unpack this. What is the actual, biological crisis these researchers are trying to solve here?
Right, so the clinical term is hepatic encephalopathy, or HEE. It's a severe complication that arises during liver failure, which you often see in advanced stages of cirrhosis. To really grasp why this happens, we have to look at how our bodies handle nitrogen, specifically ammonia.
Yes, like the cleaning chemical.
Basically, yeah. Every time you eat protein, the bacteria in your gut break it down. And one of the primary byproducts of that process is ammonia. And the intestinal wall itself also generates ammonia through this enzyme called glutaminase.
Okay, so we're naturally producing this stuff all the time.
We are. But in a healthy person, this ammonia travels through the bloodstream straight to the liver. The liver acts as the ultimate detox center. It converts that highly toxic ammonia into harmless urea, which is then just filtered out by the kidneys.
Right. But in someone suffering from HE, that detox center is essentially shuttered.
Exactly. Which leaves all that ammonia just circulating in the blood. We call that state hyperammonemia. And because ammonia is such a tiny molecule, it has no trouble at all crossing the blood-brain barrier.
Wow, so it just washes right into the brain?
Yeah, and once it breaches the brain, the real damage begins. It disrupts how the brain metabolizes glucose, it alters cerebral blood flow, and it causes these critical support cells, they're called astrocytes, to physically swell up with water.
That sounds horrible. What does that actually look like for the patient?
The neurological symptoms are grim. A patient might start with just mild anxiety or short-term memory loss, but it frequently progresses to severe disorientation, motor dysfunction, and eventually a coma.
It's essentially a toxic buildup, but, the human body is incredibly resilient. When a major organ fails, we usually have some kind of metabolic backup plan to keep the system running, right?
We do, yeah. And that backup plan is where this specific disease gets incredibly insidious. When the liver goes offline, your skeletal muscles try to step up. They try to act as a makeshift detox center.
Wait, really? Your muscles?
Yeah. The muscles literally pull that circulating ammonia out of the blood. And to neutralize it, they bind the ammonia to a specific group of molecules called branched-chain amino acids, or BCAAs.
Okay, BCAAs, I've heard of those. Like the supplements bodybuilders take.
Exactly those. So the chemical result of binding the ammonia and the BCAAs together is a brand new molecule called L-glutamine.
Okay, I think I see where this is going. If we use an analogy here, it's kind of like a sinking ship. The liver failure is the giant hole in the hull. And the crew, which is your muscles, are desperately trying to bail out the water, which is the toxic ammonia. To do that, they are scooping the water into buckets, and the buckets are the L-glutamine molecules.
That is the perfect way to visualize it. But there are two massive problems with this strategy. First, those L-glutamine buckets aren't being thrown overboard. They are dumped right back into the bloodstream.
Oh, no.
Yeah, and they eventually circulate back down to the gut. When they reach the gut, that enzyme we mentioned earlier, glutaminase, it cracks the L-glutamine right back open.
So the ammonia is just released all over again.
Exactly. The buckets have holes in them. The muscles haven't actually removed the ammonia. They've just moved it into this futile, endless loop.
God, that's frustrating. And the second problem must be the cost of making those buckets in the first place. If the crew is using up BCAAs to build them, they must be depleting a super vital resource. It's like they are ripping apart the ship's steering wheel just to build these leaky buckets. You lose the very tools you need to keep the ship navigating.
You're hitting right on the core tragedy of HE. The body cannot manufacture BCAAs on its own. They're essential amino acids, meaning we have to get them from our diet. So because the muscles are just hoarding them to trap ammonia, systemic BCAA levels absolutely plummet.
Which I'm guessing is really bad news.
It's catastrophic for the brain. See, BCAAs normally compete with a different class of amino acids, aromatic amino acids for entry across the blood-brain barrier. They use the same transport doors. When BCAA levels crash, those gates are left totally unguarded.
So the wrong stuff gets in.
Exactly. A flood of these aromatic amino acids rushes into the brain, and the brain mistakenly uses them to synthesize what we call false neurotransmitters.
False neurotransmitters. So the brain is simultaneously drowning in toxic ammonia, and it's losing its ability to send clear electrical signals because its wiring is being replaced with, like, cheap, faulty materials.
Yeah, it's a dual-front assault on cognition. And when we look at the current FDA-approved options to treat this, they are incredibly primitive. I mean, doctors primarily rely on Lactobacillus.
Which is a laxative, right?
A powerful laxative, yeah. It lowers the pH of the gut and forcefully flushes the ammonia out before it can be absorbed. But the side effects are severe diarrhea and dehydration. It makes life miserable for patients. The other main option is an antibiotic called rifaximin, or RFX.
Okay, an antibiotic to kill the bacteria making the ammonia.
Right, it's a broad-spectrum drug that stays in the gut and just indiscriminately wipes out the ammonia-producing bacteria.
But, I mean, dropping a bomb on the microbiome just breeds antibiotic resistance. Plus, based on that loop you just described, neither a laxative nor an antibiotic addresses the root metabolic failure at all. They don't replenish the depleted BCAAs, and they definitely don't stop the gut enzyme from breaking open whatever L-glutamine the muscles manage to create.
Yeah, they're just blunt instruments. They're trying to fix a highly intricate, dynamic metabolic failure with a sledgehammer. They might buy a patient a little time, but they don't repair the machinery.
Right. Okay, so just to do a quick recap here. The liver is failing, ammonia is spiking, and the body's backup plan with the muscles is actually making everything worse by starving the brain of essential amino acids. So the big question for you, the listener, is how do you fix a multi-organ failure without just bombing the gut with antibiotics? Which brings us to the actual intervention designed by the researchers at Cell.
Right, the strain engineering.
Yeah.
Yeah.
If you can't just flush the system or nuke it, you have to intervene inside the loop itself. So they decided to build a custom biological workforce to do exactly that. They started with Lactobacillus plantarum, specifically this well-studied strain called WCFS1.
Right, which is a very common, very safe probiotic. It's the kind of bacteria you ingest when you eat fermented foods like sauerkraut or kimchi.
Exactly. But they didn't just give mice a standard probiotic supplement. They utilized the field of live biotherapeutics to custom code 2 distinct strains of this bacteria, literally turning them into highly specialized microscopic factories.
Let's examine the first factory they built, which they designated LPNH3. The engineering goal here was wildly ambitious. They wanted to force this probiotic to literally consume the toxic ammonia that's pooling in the gut and use it as a raw material to manufacture those precious depleted BCAAs.
Specifically L-valine, right?
Yes, specifically L-valine.
But L-plantarum doesn't natively perform that complex synthesis efficiently. I mean, it's not wired for that. So the researchers had to give it new genetic instructions. They imported genes from 2 entirely different bacteria, Lactococcus lactis and Bacillus subtilis.
Which is such a cool approach. They grabbed a specific gene from B. subtilis because it codes for an enzyme capable of grabbing free-floating ammonia and stapling it directly onto a carbon skeleton. And that skeleton forms the foundation of L-valine.
It's brilliant genetic patchwork, but, inserting the code is only half the battle, because giving a bacteria new genes is kind of like handing a contractor a set of blueprints. It doesn't mean they'll actually build the house. Bacteria are fundamentally selfish survivalists. If they absorb ammonia and energy, why wouldn't they just divert those resources toward their own native growth pathways?
And what's fascinating here is how the researchers anticipated that exact bottleneck. Because in the initial phases, the bacteria simply didn't produce much L-valine. The metabolic flux, which is the internal flow of energy and chemical resources, was being diverted down the bacteria's preferred natural pathway.
They're just taking the energy for themselves.
Exactly. So to solve this, the scientists went into the bacteria's native genome and basically started burning bridges.
Oh, wow. They knocked out the competing pathways so the bacteria had nowhere else to go.
Yeah, they deleted 2 crucial native genes. First, the LDH gene.
What does that one do?
It normally steals resources to manufacture lactate. And second, they deleted the BCAAs, which the bacteria normally uses to break down BCAAs for energy. By removing these off-ramps, they trapped the bacteria on a metabolic one-way street. The only way for the cell to process energy and survive was to drive straight down the synthetic highway that manufactures L-glutamine.
That is so clever, but even if you force them down that highway, you still want them moving fast, right? The paper notes they optimized something called the translational initiation rate, or TIR.
Right, the TIR.
Basically, they engineered a synthetic ribosome binding site. Ribosomes are the protein builders of the cell, and by optimizing this site, they ensure the bacteria were reading those imported blueprints at maximum volume. They basically crank the factory assembly line up to run 24/7.
Yeah, so strain 1 is complete, right? A hyper-efficient machine that sucks up ammonia and spits out BCAAs. But the researchers knew that only solves half the disease. You still have the sinking ship problem.
Right, that massive buildup of leaky L-glutamine buckets that are draining back into the gut.
Exactly. So they engineered strain 2, which they called LPQ. The goal here was to act like an industrial vacuum for L-glutamine, sweeping it up before the gut enzymes could break it down into more ammonia.
And L-plantarum already possesses a native enzyme for this, right? Called GLMS1.
Yes, so the scientists simply overexpressed it. They turned the volume way up on that specific gene.
But I have to imagine there's a capacity limit here. I mean, if a bacteria is just endlessly sucking up L-glutamine, doesn't the vacuum bag eventually get full?
It does, and that creates a biological traffic jam. When the bacteria consume L-glutamine, they convert it into L-glutamate. If too much L-glutamate pools inside the cell membrane, it triggers a negative feedback loop. The cell recognizes the surplus, signals that it had enough, and shuts the vacuum off entirely.
Oh, so it just stops working. So how do you disable the off switch?
You install a drain.
A drain?
Yeah, they added what they call a sync module. They introduced another gene, ProB. This gene codes for an enzyme that it immediately converts the L-glutamate into an entirely different molecule, proline.
Okay, so it gets rid of it.
Exactly. By constantly draining the L-glutamate away, the internal concentration never spikes, the bacteria never realizes it's full, the negative feedback loop is completely bypassed, and the cell just keeps vacuuming up L-glutamine indefinitely.
Wow. Okay, let's just do another quick recap for everyone listening. We now have two highly specialized biological robots. Strain 1 eats ammonia to produce BCAAs, and Strain 2 relentlessly clears out the L-glutamine. So we've got these biological robots in a Petri dish, and the theory is totally sound. But a Petri dish is a highly controlled environment. The real question for you as a listener is, what happens when they are deployed into a living, breathing host? Like, can they survive the chaotic, hostile environment of a failing mammalian body?
And to answer that, the researchers had to subject these strains to the ultimate stress test. They utilized a very specific preclinical mouse model called bile duct ligation, or BDL.
BDL, okay. What does that entail?
It involves a surgical procedure that intentionally obstructs the bile duct. This perfectly mimics human liver cholestasis and the resulting fibrosis.
So it forces liver failure in the mice.
Exactly. The liver is severely damaged, their ammonia levels skyrocket, and the brain begins to suffer the exact same encephalopathy we see in human patients.
Okay, so the researchers administered a cocktail of their two engineered strains, which they dubbed LPNH3 plus Q, and they benchmarked it against the current clinical standard, the antibiotic rifaximin. And the biological metrics they recorded are just staggering.
They really are. The engineered cocktail completely outperformed the clinical drug. In the BBL mice, the bacterial therapy dropped blood ammonia levels by 10-fold compared to the untreated control group.
10-fold? That's massive.
Right, and brain ammonia plummeted by 6-fold. And crucially, they successfully reversed the BCAA depletion. They significantly raised those essential amino acid levels inside the actual brain tissue.
What's incredible to me is that you could visibly see the difference in the mice. Like, the treated mice didn't just have better blood work, their cognition actually returned.
Yeah, they acted better.
The researchers ran them through specialized behavioral assays. Like in the novel object recognition test, which evaluates a mouse's short-term memory by seeing if it prefers investigating a new toy over a familiar one, the treated mice showed vastly improved memory retention. They were actually remembering things again.
And they also used the open field test.
Right, which gauges anxiety, right?
Yes. It measures how willing a mouse is to leave the safe, dark edges of a box and explore the exposed, brightly lit center. The treated mice were far more exploratory, indicating much lower anxiety. The neurological fog was genuinely lifting.
But, you know, the researchers needed to prove why the brain was healing. Was it just a reduction in toxins, or was the brain actually repairing its signaling networks?
To find out, they performed transcriptomics, specifically RNA sequencing on the brain tissue.
Wait, what does RNA sequencing actually let you see?
It basically allows us to see exactly which genes are actively being read and turned into proteins in real time. It's like looking at the live blueprint of the cell's activity.
Okay, and this analysis is where the limitations of the standard antibiotic really become glaring.
Completely. Because when they looked at the RNA from the mice treated with rifaximin, the data showed that the antibiotic primarily just reduced neuroinflammation. It calmed the immune system's panic response to the toxins, which is helpful, but...
But it doesn't fix the damage.
Exactly. But the engineered bacteria, LPNH3 plus Q, they initiated a much deeper repair process. They actively restored the activity of G-protein-coupled receptors, or GPCRs.
GPCRs, those are the massive communication hubs embedded in the surface of brain cells, right?
Yes. And if the liver failure caused the brain to use false neurotransmitters like we talked about earlier, restoring GPCRs means the correct signals are finally getting through again.
Right. Because the transcriptomics showed a specific restoration of dopamine and serotonin receptor pathways. These are the fundamental networks required for learning, memory, and motor control. The liver failure caused aberrant, chaotic neuronal signaling, and the engineered bacteria just normalized it.
It's remarkable.
It sounds like using the standard antibiotic is akin to putting out a house fire with water. Yes, the flames are gone and the temperature drops, but you are left with this charred, uninhabitable frame. But the engineered bacteria, they put out the fire, and then they actually went in and rewired the electricity. They brought the dopamine and serotonin networks back online.
I love that analogy. The engineered microbes fundamentally altered the systemic metabolic environment, which literally gave the brain the exact materials it needed to rebuild its own infrastructure.
Okay, so a mini recap here. The bacteria fixed the blood, cured the brain fog, and restored neurotransmitters. But my mind immediately goes to the neighborhood they moved into. Think about it. If we send a mutant, highly optimized bacteria into a patient's gut, what happens to the native microbiome? Did they have to ruin the local ecosystem to achieve these results?
And this is exactly the Achilles heel of traditional pharmaceutical approaches, right?
Yeah.
The paper included a deep metagenomic analysis of the gut microbiomes in these mice. And when they used rifaximin, the antibiotic acted like a bomb.
As antibiotics usually do.
Right. It drastically reduced the overall diversity of the gut ecosystem. It entirely wiped out incredibly beneficial anti-inflammatory bacterial populations, specifically strains like Allostypes and Roseburia.
Which leaves the patient incredibly vulnerable to secondary infections or, chronic inflammation once the treatment stops.
But what about the engineered probiotics?
They prove to be perfect neighbors, honestly. The LPNH3 plus Q treatment preserved the natural microbiome diversity completely.
Wait, really? No disruption?
None. The abundance of those protective anti-inflammatory bacteria remained completely intact. The engineered strains did their job without displacing the critical native flora.
That's amazing. But that definitely raises a massive safety question regarding live biotherapeutics. We are talking about taking a bacteria, knocking out its native genes, inserting foreign genes from entirely different species, and creating this highly efficient mutant. If a patient swallows that, what stops it from colonizing the gut permanently? What if we want to turn the therapy off? It's a huge concern. And the FDA has incredibly strict guidelines for live biotherapeutic products precisely because of those risks. So the scientists had to engineer safety mechanisms directly into the core architecture of these strains.
What kind of mechanisms?
First, they tackled the risk of horizontal gene transfer. In standard lab engineering, scientists often use antibiotic resistance genes placed on plasmids, which are small, floating rings of DNA to help select the bugs they successfully modify.
Right, it's like a marker.
Exactly. But plasmids are easily traded between bacteria, like baseball cards. If you put that into a human, your engineered bacteria might hand antibiotic resistance to a dangerous pathogen.
Oh, that would be a disaster. So they had to remove the plasmids entirely.
Yes. They use a technique called plasmid curing.
Plasmid curing?
Basically, they integrated the synthetic therapeutic genes directly into the bacteria's main chromosome, locking them firmly in place, and then completely purged the plasmids from the cell. So there are 0 antibiotic resistance genes left to share.
That's super smart.
And secondly, they had to ensure the bacteria wouldn't breach the gut and cause a systemic infection. They fed mice high doses of these engineered strains every single day for 28 days.
And what did they find?
Testing showed no dangerous spread to organs like the heart or the spleen. The minuscule fraction that did cross the intestinal lining was immediately and quietly cleared by the immune system, without triggering any systemic inflammation.
Okay, so it's safe systemically. And regarding their permanence in the gut ecosystem, I mean, do they stay there forever?
The data on that is perhaps the most reassuring part of the whole study. Once the researchers stopped the daily dosing, they tracked the engineered bacteria in the mice's feces. Within 72 hours, the therapeutic strains dropped completely below the limit of detection.
Just 3 days.
Yeah. They simply do not have the evolutionary adaptations to permanently colonize and compete in a native gut. I like to think of them as temporary punch-the-clock workers.
Punch-the-clock workers.
Right. You ingest them for a shift, they execute the heavy lifting of processing the ammonia, and then they clock out and wash completely out of the system.
Wow. Okay, so what does this all mean? Let's bring this entire narrative together. Scientists were faced with a cascading biological disaster. Lethal ammonia spikes, completely depleted BCAAs, and a toxic L-glutamine loop that was driving a failing liver and a dying brain. Instead of trying to medicate the symptoms with blunt chemicals, they built a customized two-part bacterial workforce.
Yeah, strain 1 was programmed to consume the poison and synthesize the exact amino acids the brain was starving for.
Right, and strain 2 was programmed as a relentless vacuum to break the toxic loop. By deploying them into a failing system, they provided a metabolic bridge that effectively acted as a surrogate liver, rescuing the brain's cognitive networks in the process.
And they achieved all of this safely, preserving the patient's native microbiome without leaving a permanent artificial footprint in the gut.
Exactly. To everyone listening, your own gut-liver-brain axis is actively operating inside you at this very second. We historically think of our microbiome as a static, passive passenger, maybe something we occasionally support with some yogurt or fiber. But this research proves that the microbiome is actually a highly dynamic, programmable operating system. When the host's primary hardware starts to fail, we now have the technology to literally patch the system with new, living biological code.
It really brings us to a profound threshold in medicine. And I think it leaves us with a provocative question to consider.
Let's hear it.
If we can program a common, everyday probiotic to efficiently bypass a failing liver and protect the brain from neurotoxins, what is the limit of this technology? Think of the countless chronic, supposedly incurable, multi-organ diseases we currently battle. Are we entering a new era where, instead of taking a daily chemical pill with a laundry list of toxic side effects, we simply swallow a custom-coded bacterial workforce?
That is wild to think about. Are we moving away from static chemical interventions and toward an age of temporary, highly targeted biological software updates?
It really seems like we might be.
It certainly makes that minor brain fog after a heavy meal feel like just a tiny glitch in an incredibly complex machine, a machine we are finally learning how to actively reprogram. Thank you for joining us on this deep dive. Keep exploring the microscopic worlds within.